So we're just going to try and wrap up what I wanted to discuss, and because it's clear that everybody's coming from very different backgrounds and baseline levels of understanding, I want to make sure that everybody's very, very clear, otherwise you'll be very confused the next few days. So as you were saying earlier, mutations are, can you pull up this board? This one we can keep. So as we said, the source of new genetic variation is at some basic level mutations. These can be in the form of point mutations, which you already discussed a bit in the past few lectures. Insertions, which will be new bases are put in between, or deleted, which will be deletions. You can also have inversions, which are chunks of DNA which get flipped, okay, due to errors during replication or due to recombination. You can have translocations, which will be movements of chunks of DNA somewhere else in the genome, or duplication, as the name suggests, a piece of DNA gets duplicated, right? And all of these and more weird combinations of these can be serving as mutations that give rise to variation. In fact, we think of anything that changes what's there in the sequence context, and doesn't have to be coding DNA necessarily. We'll talk a little bit more about this in my next set of lectures. And then we already talked about migration a bit as well as recombination, right? So all of these can introduce new variation in a population, and the whole point is to ask how much of this variation is there, what generates it, what maintains it, and what selects on it. So how much variation do populations actually harbor? What are some ways to measure it? Anybody have any idea? How would you measure how much variation exists in populations? Any population of your choice. Sorry? Analysis of variance, what does that mean? Right. So you choose one bird population or some plant, whatever you're interested in, right? And you want to quantify somehow how much variation exists in that population, right? How would you do it? Yeah. Looking at phenotypes, okay, so that would quantify phenotypic variation. Randomly sample and do a genetic analysis. What kind of genetic analysis? Okay, so you want to start sequencing a little bit. How much do you want to sequence? Genomes. I mean, why not? But why? Yeah, so it's not like there's one answer to this, right? And that's the point I really want to get across, is that there isn't one answer to these questions, and we want to quantify how much variation there is. You also want to think about what kind of variation do you actually care about? Do you care about variation that's neutral? Do you care about variation that is potentially under selection? Do you care about variation that has evolved under selection in the past, may or may not be under selection? What is it that we actually want to measure, right? And the answers to these questions will really change how you're going to go about it, right? It may be sufficient to measure phenotypic variation to get an idea of underlying genetic variation in some cases. It may not give you a good idea in some cases, right? It may be sufficient to measure some set of genes to get an idea of diversity. It may not be. Again, depends on what you're asking. And so keep these things in mind as you're thinking of your projects and thinking of your own real life projects for your work as well. How much of this variation is relevant for evolution? How much of it is relevant for adaptation? And what does this depend on? And the hope is that during the course of the next few days, you'll at least get the tools to think about it and have an idea of what factors are important to be able to answer these questions, okay? And so, like I said, this is just something to think about. So I want you all to now play with some simulations. There's an online population genetics simulator. The link is in Slack as well. I've just posted a little bit ago. So open up your laptops. It's a very easy, simple, and nice way to get some intuition about some of the things that Kavita has already spoken about earlier. What is going to happen to alleles in populations. Okay. So explore this a bit, and I want you to particularly try and answer some questions. Okay, everybody has the page open, right? So let me also navigate to that page. Okay, so this is what it looks like, right? And, oh no, what did I do? Okay. Okay, so here you have buttons to toggle population size, number of generations for which your simulations are going to proceed, and red allele starting frequency, okay? So you're going to change these numbers and then run the simulation, all right? Dial up and down, run the simulation. You can also change how much selection is acting on different alleles and whether there are mutations happening to and fro from the two different alleles. So we'll ignore these mutations for now, but just playing around with these three things and thinking about stuff, I want you to find 3 ways in which you can make the red allele fix. There are 2 alleles in the population, red and blue, okay? You already know some of these answers just intuitively. Anybody? How do you make one allele fix in the population? What would you do? Speak up. Increase the starting frequency, good. So if your starting allele frequency is higher, the red allele is more likely to fix in the population. Everybody agree with this? Yeah? All right, somewhere from the middle. Another way? Yeah. Increase the mutation rate to red. All right, that's cheating, but sure. Okay. That's one way to help the red get fixed. Increase the fitness of the red allele, excellent, right? Or other way around, reduce the fitness of the blue allele. Good. Any other ways? No, hang on, you already spoke. Somebody hasn't spoke. Backbench. Awake? Yeah? Come on. Use those brain cells. Any other ways? In this simulation, is there any other way to get to this? No? Sorry? Increase the population size, good, right? So if your red allele has a benefit already, right? If you increase the population size, the probability that it will get fixed is going to be higher, right? Because selection has a chance to be more efficient in that population, okay? Good. Any external factor you can change here? Cheating.
Just increasing population size won't make red.
Not by its own. It has to be when red already has an advantage, right? Yeah. And if your initial size was small, correct? Okay. One more, quick. Cheating thing which doesn't exist in this simulation. Simulate from one number of generations, that won't necessarily help. Again, it's going to help only if you have, well, no, that won't change the probability of fixation, right? That we already talked about.
A higher growth rate for red.
Same thing as selection is stronger, favoring red. Yeah? Survival chance is there already. We talked about selection on red. Sorry? Oh, that's serious cheating. Okay. Okay. How about we bring in a lot of red alleles from migrants, right? That's not in the simulator right now, but that's something you could do. Okay? Good. So now another question, and I want you to do this on your own a bit. So set the survival rate so there's a heterozygote advantage. Okay? Can you do that in your own simulators? What happens to the red and blue alleles? So I want somebody to do that and then tell me what you find. Sorry? You see coexistence, correct? Do you see that? Yes. Okay. Can somebody find a way to break that expectation? Where you have heterozygote advantage and yet one allele can go extinct. Is that possible to do? Population size is very tiny. How tiny is your population size? 20. And which one goes extinct?
Blue is going extinct.
Okay. Everybody agreeing with this? Should this happen? Or has she done some cheating somewhere? Okay. Blue goes extinct. Okay, population size being small. All right, good. Set the blue allele to have a selective disadvantage. Okay. Can you change anything else to prevent its loss? Because you expect it will be lost eventually. How much disadvantage have you set for the blue allele? 80%, okay. And the red allele has 100% survival, okay, good. Then, does the blue get lost? All right, so what can you do to prevent that loss? Increase mutation rate, okay. Okay, what else? Heterozygote advantage, good. What else? Which will just prolong the torture, yeah. But yes, sure. It will delay the, well, the torture is like you, okay. What else, anybody? What about population size? Reduce the population size, right? Yeah, in some instances, depending on how low your population is and how big is the disadvantage to the blue, you might see some fixation of blue due to drift, right? Okay, good. So these, so if you want to write down these questions or just play around with it on your own later, okay? All right, so the core idea here is that selection, the selection coefficient as well as effective population size together determine fixation probability and dynamics, right? One alone is not sufficient to understand what's going on completely. You need both, and it's the combination of both which is important. When n is small, drift is going to dominate, and S has to be very large for a beneficial allele to fix, okay? So an S of 0.002, which is less than a 2% advantage, is not going to cut it, all right? If your n is very, very small. Because the chances that a beneficial allele will be lost by chance are going to be higher. At large n, selection dominates, and even a mildly beneficial allele can be fixed at a large population size, okay? Is everybody's intuition on the same page, or should we discuss this a bit more? Bit more? So when population size is very large, selection is very effective, right? So what that means is chance plays a smaller role. Okay, so I suggest you play around with these simulations a bit more today evening. Try it out on your own and you'll see what I mean. Test what I'm saying. Don't believe me just because I'm saying it, okay? So please do that. Even at large population size, not all alleles behave as if they are evolving under selection. And this is a very important point, and it's the basis of what Kimura and King and Jukes proposed as the neutral theory of molecular evolution. Okay? So they're saying is neutral alleles can also behave as if they are evolving under drift, and even beneficial alleles under specific conditions, under small population sizes, will evolve as if they are under drift. Okay, that S value doesn't matter very much when your population size is small. And this is partly why they were proposing that a lot of genetic variants that we observe in natural populations is behaving neutrally. Okay, from a completely fitness point of view, it may not be actually neutral. It may be kind of mildly neutral or mildly deleterious or mildly beneficial. But it's going to be more likely to behave as if it's neutral if the population size is not infinite, okay, or not very, very large. This is important. Again, think about this a bit, play around with your simulator, and you'll get a sense for what and why is going on. We can discuss this more later. But it's important to remember that our intuition about what should happen to an allele given its S value is not always correct, okay? All right, so now let's go out to, all of this is saying, okay, this S value, and where does all that come from, right? How do we measure this? So we come to what's called the distribution of fitness effects. It's a statistical distribution. And this is effectively telling us if I pull out random mutants, okay, or random alleles from a population, what is going to be the average effect of such alleles? Is variation that we see in populations largely neutral? Is it beneficial? Is it deleterious? What are the effects? Okay, and these statistical distributions are useful because they are what get plugged into models and theoretical theory and simulations that ask what happens at time scales and at population sizes that experimentalists cannot address, right? So to make any predictions, we have to use theory, but the theory has to be based on reasonable data somewhere. So what are these data looking like? I'm showing you here data from vesicular stomatitis virus. So this is a virus which infects plants, and what they've done is they've made random mutations, okay, in some important gene or maybe multiple genes of this virus, I forget. And they've allowed these different viral particles to try and infect plants. And they've asked how well have all these different viral particles performed when they are allowed to infect a plant. So they're asking how many progeny or offspring virus particles are being produced by each of these mutants. What you see here is on fitness on the x-axis and the frequency of a particular fitness value on the y-axis, right? So it's some large collection of mutants. A fitness effect of 1 is basically going to mean that it is neutral. So this is relative fitness, okay? So relative fitness of 1 means it's neutral alleles. 0 obviously means lethal, right? So these are virus particles that completely failed to reproduce at all. And you can see that's the bulk of mutations here, right? 40% of all mutations that they tested in this virus were lethal. So what does this make this distribution? More or less bimodal, right? So you see one peak at the lethal point, you see another peak at the neutral or nearly neutral point, almost neutral, sort of close to 1. And then there's a whole bunch of deleterious mutations here to different degrees, and a few beneficial mutations here which are weakly beneficial, okay? So this is what a DFE looks like from some data, okay? And this kind of DFE people have wanted to generate for a lot of different taxa because certainly the DFE is not going to be a constant. That's one God-given thing, right? That's true for everything. So what do we know about DFEs? What is the phenotypic effect of mutations? People have tried to model the DFE using many different kinds of distributions. The gamma distribution has been used a lot. And this distribution is kind of nice and it's useful because with a few tweaks to the parameters that define its shape, you can get distributions that look very, very different, right? People have also thought a lot about the distribution of the DFE, only focusing on the beneficial side. Why? Because that's relevant for adaptation. In the past, people actually used to focus only on the deleterious side. Why? Because they were worried about load, okay? That if you have a lot of deleterious mutations in the population, it's going to drag fitness down, and that people were obsessed with. Then people became obsessed with beneficial mutations, okay? And now people are obsessed with the whole thing. Let's define the whole thing, okay? Let's not focus on only one part. So under adaptive evolution, there's this one mathematical way to think about this called the extreme value theory, based on which people predicted that that far end, the tail end of the beneficial side, which is the right side of a DFE typically, is going to look exponential. Now, why is that important or relevant? It makes the mathematics a little bit easier to work with, okay? And it also means that there's going to be an exponential decline in how much fitness benefit you're going to get and how many beneficial mutations, high value beneficial mutations is the population going to have access to, right? And what that basically means is things are hard in terms of adaptation, right? There are very few fantastically beneficial mutations that populations may have access to. That's what all of this is suggesting, right? So this was kind of important to know and to measure. These are all cartoons, by the way. These are not real data or something, okay? And people for a long time were trying to get data and ask, the data we have, are they fitted well by this distribution or some other kind of distribution, and so on and so forth, so that the modeling can be done in a more realistic way, okay? So what do the data actually look like? So people have been then trying to measure. It's hard to do these experiments because you need to measure for single mutations, right? what this DFE looks like. You need to acquire single mutations. Imagine doing that in an organism like a horse. Not possible, right? I mean, I don't think anybody's even dreamt of doing horse DFEs. I have no idea why I said horse. But you can do this in bacteria reasonably well. You can do this in viruses, right? You can do it in cell lines and so on. You can do it for single genes where you can make random mutations at a high throughput way. That is everything that people are doing currently in the last 10 years. There's been huge explosion of how many studies do this kind of work, okay? But we're still limited by data in the sense that it's not very widespread. It's very narrow, restricted to a few taxa, few kinds of environments where we can measure these things reasonably well. So what do the data look like? I want to show you a few different kinds of information. Here is people who made about, 10, 12 years ago, people who made mutations in the ribosomal protein, which is a very important protein in bacteria, makes part of the ribosome, so you can't do translation without it. And what they found is in Salmonella, most mutations are deleterious, as you might have expected, especially for an important gene like this, which has presumably been optimized for its function over evolutionary time. What was interesting, so by now hopefully you can read DFEs quickly, right? So the red line is showing you the 1 point, what indicates neutral mutations, and pretty much everything is on the left of this, right? So everything is going to be deleterious. They don't see that lethal peak, presumably because here they would not have these strains grow. This is not like viruses where you know what particle you have and then you try and infect it, right? This is bacteria where the mutation may have existed, but that bacterium was dead. And so you have no idea what you've missed. So the lethal mutations are missing. The DFE is truncated. What's interesting is that these are data for LB, these are data for M9. Now, LB is a rich medium, M9 is a poor medium where growth is relatively restricted, and the DFEs look actually more or less similar, right? There's some differences and one can discuss that, but this speaks to the idea of how much impact environments can have on the DFEs. And we talked about it earlier that your fitness or your phenotype is not determined only by your genetic makeup, it's also dependent on your environment, right? I mean, and you all can figure this out or have examples of this very easily. Perhaps you were the shining star of your elementary school, right? Always first in class or whatever. And then as you grow, it becomes harder and harder to be maintaining that status. If you went to a very competitive undergraduate or master's or Ph.D. program, there are a lot of smart people around you, right? So it depends on the environment how well you're going to do also. So we also wanted to measure the DFEs, and this is work by Mridula, who was a Ph.D. student in my lab. And so we collected a bunch of random mutations that occurred during what's called mutation accumulation in the lab with E. coli. What is mutation accumulation? You've heard of selection experiments, right? You can experimentally evolve populations of insects or bacteria, horses if you want, to evolve under some selection pressure, right, that you're imposing specifically. What you can do is that you can skew the DFE that you're getting out of those kinds of experiments, because you're going to, of course, choose selectively beneficial mutations a lot more. So you're missing the whole left side of the DFE quite a bit. So to avoid that, we wanted to allow for selection strength to be very weak. And so we use this paradigm called mutation accumulation. Effectively, what you do in this is you take single colonies which you're choosing day to day based on a pre-marked spot on the plate. This is an agar plate. That's a bacterial colony, okay? And so you keep doing this where this colony gets transferred and spread out. There are other ones, but this is the chosen one, okay? And so what effectively happens is that you get single cell bottlenecks, right, repeatedly. And that reduces the strength of selection, magnifies the impact of genetic drift, so that any mutation is now not competing with others more or less, right? So let's imagine going from here to here. Each colony is formed by a single original bacterial cell, okay? Now this guy here, let's imagine, happened to get a mutation between growing here and going here, right? That mutation, unless it's lethal, I'm going to pick it up. I'm going to sample it because I already said, when I move that when I spread that colony over here, before anything grew, I decided that was the spot from where I'm going to pick a colony, no matter whether it's small or big or yellow or red or whatever, right? Well, I should be worried if it's not E. coli, but anyway. So that's what sort of, basically we're reducing selection as much as possible, allowing drift to, and so then we get a better sample of random mutations that could accumulate, okay? So that's what Mudala has done here. And then these are DFEs, again you're used to seeing the DFEs. And then what's interesting is she had this collection of 80 mutations that she sequenced the genomes for to identify a single mutation in the wild type E. coli background that accumulates over this sort of experiment. And then she asks, what impact does it have on growth rate, okay, of the culture? And that growth rate we're using as a proxy for fitness. So red line in between is showing you what's corresponding to neutral. It corresponds to 0 on this x-axis because of the way we're calculating growth rate or fitness here. But just believe me when I say that it's 0 is where neutral mutations are. And so what you'll notice is that in different media where she tests the same set of mutations, the DFEs look a bit different, right? In some cases, like in glucose and trehalose, they look a lot similar. There's a lot of mutations that are clustered around neutral. There's some lethal mutations that we're able to catch. This is because these guys were evolved on a rich media, and then they were tested on other media. So in that case, we can actually identify lethal mutations, right? These guys, some of these, about 4 strains, failed to grow on these other media. So we know they're lethal in those media, but clearly they were not lethal on LB, the rich media where they were evolved originally. You can also see that in lactose, there seem to be more beneficial mutations. In galactose, there's a whole bunch of nice beneficial mutations, right? So the DFE does vary across environments, and this is observed by other people as well. So in contrast to what you saw for the ribosomal protein DFE, where it did not vary very much, most other cases people have tested across different environments. They found that these kinds of random mutagenesis or across the genome or across single genes, you do find big differences across environments. Yeah.
On the left side, we have more kind of lethal mutation.
The left, the extreme left is lethal. All of this is deleterious in between. Make sense?
In the previous slide, like you mentioned about the red bar,
Uh-huh.
and then at 0.678, so those are the lethal one, right?
No, these are not lethal still. These are still deleterious guys. Lethals could not be assayed in this kind of assay.
So those are deleterious ones.
These are deleterious, yes. Any other questions so far? Everything clear? Yeah? All right. So I want to then also go to phenotypic effects of other kinds of mutations. These were all single mutations that we were discussing so far. And what is the distribution of fitness effects for those? What about big kinds of mutations like ploidy changes? Okay. I think many of you probably are familiar with the idea that ploidy changes a lot. So this is a diploid plant. This is, I think, Arabidopsis, but maybe I'm wrong. It's 2x, okay? And then there are strains or varieties which are 4x. So there's been a duplication or there's been hybridization. This is very common in plants. Ploidy is nuts in plants. And it's interesting because it has all these very interesting phenotypic effects, right? So you can see clearly that when ploidy increases, the structure of the leaves changes, the size of different parts of the plant increase, typically with ploidy. This is observed in many other plants as well, okay? Here's a case in humans where ploidy has an impact on disease. So if you have Down syndrome, this is a situation where chromosome number 21 fails to segregate during meiosis, and as a result, there's a triploid condition of this chromosome, and babies born with this have various kinds of developmental defects and problems, and they don't usually survive for very long, right? So there are very big effects of ploidy changes, even with single chromosomes or with multiple chromosomes. A lot of really interesting work in evolution focusing on what happens when ploidy increases, because now dosage of all genes has increased, right? Or could increase in the cell. How do cells deal with this and so on? So it's really interesting stuff. Okay, so here are some other examples of mutations that alter phenotype. This is work from actual natural populations and so on, and you'll hear some more examples from Krishnameg in his talks later. So this is a famous insect that's been studied a lot, There's two forms of this. I know this is a bit difficult to see, but this one has a stripe in the middle. It's a white stripe. And this one does not have a stripe, okay? So these insects live on plants and hang out on plants. And what people have shown in many years of work is that these guys will usually hang out on a different plant, okay, next to each other. And this stripy plant has stripes on the leaves. That's where the form with the stripes will hang out. And this one is better camouflaged on plants without any stripes on them, okay? The leaves don't have stripes. So they did a bunch of genome sequencing to try and identify, what mutations are underlying these phenotypic differences. And what they find, okay, you're going to see examples of these kinds of things much later. I think Shweta will probably talk about this a lot afterwards. What we're showing effectively here is on the bottom, on the x-axis, is position along the genome, okay? Imagine this is the entire genome of this thing scrunched up. And on the y-axis is some measure of saying how many differences, how much polymorphism do we see at that location that is associated with the phenotype that we're seeing. So higher the values, higher the likelihood that there is some causal mutation in that region, okay? Again, the details you will hear later, but for now this is sufficient. And what you can see is across these different parts of the genome, okay, each little bunch here is a chromosome. Many of them are kind of bobbing up and down within some average value, right? What one is looking for is big outliers, which means that there's a lot of differentiation at that locus between these two forms. And you don't see that in this by and large. So what they conclude from this study is that in this case, this phenotypic difference is probably caused by lots of different loci, lots of different mutations at many different parts of the genome. Okay, there aren't these single big beneficial mutations that have allowed for this phenotypic difference. All right? So what are the general patterns that people find? In cases like aphids, there's a continuum of differentiation. Sometimes there are few changes, sometimes there are lots of changes. In lizards, there are bursts of differentiation that happen. So lizards in some parts of the world seem to suddenly acquire a lot of mutations, lots of phenotypic diversity. In butterflies, there's color pattern changes, courtship song changes. In crickets, and the genetic architecture is different in these cases. There are a few genomic peaks in crows, genome-wide differences in herrings, lateral plates in sticklebacks, flowering time in maize. These are all cases where in some places you have one or few loci of large effect, multiple loci of small effect, okay? So these cases are all across the spectrum. And so it's not, we don't generally have an answer to the question of is adaptation or evolution driven by big jumps, okay, a few big mutations that are to the right side of our DFE, extreme right, or are they driven by lots of tiny mutations closer to the neutral, okay, close to the right side of the DFE. Make sense? The answer is that everything is possible and everything is observed. So the more important question is under what conditions will you observe one or the other? And that again is where theory plays a role in making predictions which can then be tested. Yeah. Yeah. What are the changes that underlie these big changes that we see? I mean, what people mean by genetic architecture is all kinds of things, but yes, roughly that's the idea. Is it few loci, many loci? What kinds of mutations? Big structural mutations or small point mutations? Yeah.
Let's say there's an individual, he or she has genes which help them in producing better muscles or something.
Better what?
Better muscles.
Muscles, huh?
But they did not get good nutrition during their childhood. And so they're not developed. So epigenetic changes take place and these are permanent.
Why do you have to invoke epigenetic changes?
Because they didn't get good nutrition during childhood. They will not develop good muscles.
During childhood, yeah, but that doesn't mean that if they can't have good nutrition later on, they can't develop good muscles later on.
They will not have good nutrition later also. So they will not have muscles. And if having muscles is selection, then though this person has that variation,
Yeah. Yes, can be lost. So the question broadly is, what about your genotype predisposes you towards having a high benefit or a high fitness, but your environment conspires against you, right? And makes it so that that outcome at the phenotypic level is not going to be happening. What then? Too bad. That's what, really, yeah. So this just goes back to the idea that your genotype does not predict accurately always your phenotype and your fitness always, right? There's a lot left to a lot of different factors that go between you as a little blob and you as a individual adult who might or might not have any fitness. Yeah. Okay. Something else I meant to say. I feel like it was super important to clarify. Okay, never mind. Okay, so that's where we're at. I feel like I should stop here. Yeah, we'll talk about, okay, maybe this is useful. So what generates phenotypic variation? Plasticity. Environmental change alters phenotype, despite we kind of went over this already. Like it could be deterministic or it could be stochastic, which would involve noise in transcription factors or translation. Yeah, I think we'll just stop here for today. I'm done as well. And if some of these topics come up later, we can bring them up later. But the most important part was to go through some ideas of what's happening with NES. Please play with the simulator on your own and find me tomorrow if there's any questions. All right? Cool.
In fact, please do go through that simulator because I'm going to discuss the same NU and NS. So great.
Okay, and then, yeah, just a couple quick things about the projects that you're supposed to be doing with your group. So hopefully everybody's found their group members.
Actually, can we identify? Is there a one-to-one? Has everyone found their project? Can we just go through that now?
Yeah, sure. Yeah.
So,
have you identified your groups first?
So let's go through the list. Group 1, have you found your project? Okay. What's your project, group 1?
Have you decided your project?
No.
Okay, it's okay, you can put it on Slack then. Group 1 can identify, yeah, they can put on Slack.
Channels specific to our tables.
Sure, if you want, I can make those channels. I can actually just make the channels and you do what you want with it, yeah. So I'll make
Can we pick any of the topics or is it like already mentioned about one?
No, you can pick any topic, but you need to discuss it with your group members and decide a topic, right? Ideally, we'd like all 8 questions to be addressed by different groups. But if you're like extremely anti one question, fine, 2 groups can do the same topic, right? The point is not to choose something that you already know something about or are comfortable or happy or whatever. The point of this is to do a group exercise, talk to each other, okay, learn from each other, and think about a project. So what do we want from you in the end? Kavita, please jump in if yeah. So we want you to work together. The questions are such that they are very broad and general, okay? So for example, maybe one question says, how do you find out which pathogen is infecting some rabbits which are dying, okay? I just made this up right now. But what I want you to do is not to tell me exactly what PCR you're going to do, okay? What we want from you is to say, okay, how are you going to approach this problem? Are you going to do experiments? Are you going to do observations? Are you going to use phylogenetics? Are you going to use theory? If you're going to use theory, what kind of modeling would you like to do? What parameters might be important? If you're going to do experiments, what kind of experimental design are you thinking of? What is your sample size? What is your model organism? Where will you conduct the experiments, as in laboratory or outside, all right? If you're going to go collect data from the field, what kind of data would you collect? And all of this has to be connected with the question. You have to be justify what you're doing. Okay, you can't just randomly say, I'm going to spend $1 million and I'm going to go sample from across the world's oceans for something or the other. It has to be relevant to your question. Okay, so justify it, think about it, come up with a concrete plan. And on the last day, we have 2 slots for student presentations. There's 15 minutes for each group, which means you have 10 minutes to present. You can choose to present as a group. So you take turns to present, slides, whatever you want, chalk, board, or one of you can be a representative or 2, whatever you want, up to you. but take 10 minutes to explain how you want to approach that question, how you propose to solve it, and if possible, problems that you can encounter, right, that you foresee having encountered. And then think of how you would approach those problems, okay? So think of it really as a proposal for work, and you have to defend it, you have to come up with a plan. And in the process, hopefully, you'll be able to think more about what you're learning in the course. All right? You're welcome to discuss this whenever you want with your group members. You can stay back after the classes are over during the day. You can talk at lunch, up to you, whatever you want. Any questions? Cool. Enjoy. Also, please read the journal club papers as best as you can. We don't expect you to understand everything in those papers. That's fine, but give it a shot, okay? Because it's useful if people come having read some of these papers so that you have some idea. And we're not sitting here explaining the details. We would like discussion to be at a slightly higher level than that, all right? Okay, see you tomorrow morning.